Hello everyone, I would like to share my new article publishing in iMeta. The title is Synergistic Effects of Diazotrophs and AMF on Soil Biological Nitrogen Fixation after 3 decades of fertilization. Biologic nitrogen fixation via diazotrophs is an important ecological process for the conversion of air nitrogen to biologically available nitrogen. Although symbiotic pathway contributes more biological nitrogen fixation than a symbiotic pathway, a symbiotic nitrogen fixation occurs in the soil, litter layer, and even stalk. However, owing to the fact that nitrogen uptake directly from the soil is less energetically expensive than the biologic nitrogen fixation, large amounts of inorganic or organic nitrogen inputs challenge the contributions of biological nitrogen fixation. AMF are well known to bear numerous advantages to their host plants, such as improving nutrients uptake, increasing plant tolerance, and stimulating carbon and mineral nutrient cycles. AMF can also play an important role in modifying biological nitrogen fixation in the plant size-sphere and height-sphere. A recent study reported that synergies rather than antagonism between AMF and the diazotrophs prevail in the interspecies relationship, which may improve soil biological nitrogen fixation acquisition. However, most studies have neglected the underlying implications of the interactions between AMF and diazotrophs. In this study, we used a long-term field experiment set up in 1988, which included typical fertilizations in China, such as the chemical fertilizer, cow manure, white straw, and green manure. We examined the relationship between soil biological nitrogen fixation rates and diazotrophs and their helpers. We hypothesized that the first one, long-term fertilizers, would inhibit the soil biological nitrogen fixation and alter the diazotrophic community structure. The second one, compared to the entire microbial community, unique occurring ecology cluster of diazotrophs and AMF might predict the biological nitrogen fixation rates better. The last one, long-term fertilization would lower the synergistic relationship between diazotrophs and AMF. The biological nitrogen fixation rates were promoted relative to the non-fertilization control. Long-term organic fertilization significantly increased the absolute abundance of diazotrophic and AMF communities. The absolute abundance of diazotrophs and AMF presented strong and positive correlations with the biological nitrogen fixation rates. We then evaluated the effects of the long-term fertilization on the community structure of diazotrophs and AMF. Long-term fertilizations remarkably shifted the community structure of diazotrophs and AMF. For instance, long-term organic fertilization, especially green manure treatment, significantly increased the relative abundance of Skermanella and Azospirillum. We then built a correlation network used the dominant diazotrophic and AMF ASVs and found 4 ecological clusters. Long-term fertilization significantly increased the relative abundance of module O and 3 and reduced that of module 2. The green manure treatment had the greatest enhancement in module O. We also calculated the number of positive relationships between diazotrophic and arbuscular and mycorrhizal fungal species and found that high soil fertility caused by organic fertilization reduced the synergistic relationship between diazotrophs and AMF, which supported our third hypothesis. However, green manure treatment could reverse the decline with module O. We further calculated and compared the differences between observed and expected phylogenetic diversity for each module, and found that there were phylogenetic overdispersions for diazotrophs in module 1 and the long-term co-manure and the white straw regimes, and for AMF in module 2 and the non-fertilization regimes. This finding suggested that long-term co-manure and white straw may have been selected against the diazotrophs associated with module 1, while long-term fertilization selected against the AMF associated with module 2. Among these modules, the relative abundance of module O was positively correlated with the biological nitrogen fixation rates, while that of module 2 was negatively correlated with the biological nitrogen fixation rates. In module O, most diazotrophs belong to the class alpha proteobacteria that are often classified as the co-occurrence populations. This finding implies that the co-occurrence diazotrophs still maintained the ability of biological nitrogen fixation, which would dispel our previous knowledge that the environmental resource richness would reduce the biological nitrogen fixation ability. Unlike module O, the diazotrophs with module 2 was the loser and the fertilization regimes. Our partial least square path modeling further showed the long-term fertilization directionally select certain co-occurring ecological cluster of diazotrophs and AMF, rather than their entire microbial community to promote the soil biological nitrogen fixation, which supported our second hypothesis. In summary, this study provides robust evidence that long-term fertilization increased soil biological nitrogen fixation rate, and green manure treatment showed the highest enhancement. The increase in diazotrophic and AMF absolute abundance and the relative abundance of the key ecological cluster module O stressed soil biological nitrogen fixation. Additionally, green manuring can reverse the decline in the synergistic relationship between diazotrophs and AMF with module O, and this had the positive correlation with the biological nitrogen fixation rate. In this study, we highlight that the soil biological nitrogen fixation was affected by fertilization through increase in diazotrophic and arbuscular mycorrhizal fungal absolute abundance and the relative abundance of the keystone phylotypes, and the interspecies correlation between diazotrophs and AMF with key ecological cluster. Among the 4 fertilization regimes, green manuring is a more effective strategy for improving soil biological nitrogen fixation rate.